and respect to our today's speaker, Professor Dr. Kumar Pal, for giving us his valuable time for our student as well as for our webinar too. Now I am going to introduce Professor Dr. Kumar Pal to all participants that today's our lecture of Professor Dr. Kumar Pal. He is an eminent professor in agriculture biochemistry in Bidhan Chandra Krishi Vishwavidyalaya. Professor Pal has authored and co-authored multiple peer-reviewed scientific papers and presented works at many national and international conferences and have claimed recognition from honorable subject experts around the world. Now, Dr. Professor Pal is actively associated with different societies and academies in India, not only India, over the world. The academic career is decorated with several reputed awards and funding. Dr. Professor Pal research interests include crop husbandry, crop nutrition, natural resource management including water management research, best management practices along with physiological aspect of crop. Now I would request to Professor Pal to start your lecture, sir. Please, sir.
Okay, thank you very much. Actually, good afternoon to everybody. I am really privileged to share my some views about this well-known topic, that is regulation of Calvin-Benson cycle. Can you see the slide? Hello? Hello?
No, sir. No,
it is not visible, the slide?
No, slide is not visible.
Please, sir, share your screen.
I have already shared my screen. Okay, just wait a bit. It is visible?
No.
Hello?
No, not visible.
No, sir.
Okay, just. Now it is visible.
Yeah.
Yes, visible.
Okay. Okay. Okay, good afternoon to everybody. Actually, today I would like to share some views about the regulation of Calvin-Benson cycle by light. And this, we are familiar with this photosynthesis since our school days. So this photosynthesis, as we know, it is almost all plants are capable of photosynthesis. This is a light stimulated process that consumes carbon dioxide and evolves oxygen. Actually, as a matter of fact, this photosynthesis, it encompasses both a complex series of reactions involving light absorption, energy conversion, electron transfer, and multi-step pathway that convert this carbon dioxide to carbohydrate. Actually, most photosynthetic organisms, they convert this light energy to the stable chemical products according to this equation. So this equation signifies that photosynthesis is a biological oxidation reduction process, where this carbon dioxide serves as an electron acceptor. Water is a reducing substance which serves as an electron donor, and CH2O represents the carbohydrate which is formed by the accepting electron by carbon dioxide, and oxygen, that is the product formed by the oxidation of the water. So if we consider this biological oxidation reduction process, it is accompanied by the transfer of electron. So to see more clearly this transfer of electron, this oxidation reactions can be broken down into half reactions. In this first reaction, the water is oxidized to give 4H and 4 electron. This carbon dioxide takes up this 2 electron and 2 proton. It is converted into the carbohydrate CH2O and half oxygen. The rest of the 2 electron and 2 proton, they are accepted by this molecular oxygen to give the water. Now, there are two types of photosynthesis. One is the oxygen evolving photosynthesis. Another is the anoxygenic photosynthesis, in which other substances other than water serves as an electron donor. For example, this purple sulfur bacteria, they use H2S as the reducing compound, and instead of forming oxygen, they gives the elemental sulfur. But most interestingly, this oxygenic photosynthesis was an evolutionary innovation of cyanobacteria that led to the accumulation of the oxygen in the Earth's atmosphere more than 2 billion years ago. So this is a very significant, but photosynthesis not only providing the carbon, this dry matter, biomass or fossil fuel, it gives oxygen as a product which is required for the respiratory activity of the multicellular organisms and many unicellular organisms. And this oxygen evolving photosynthesis, it is an evolutionary innovation of cyanobacteria that led to the accumulation of this oxygen in the Earth atmosphere more than 2 billion years ago. Actually, before we go into the actual topic, I want to give some overview, because during the 1950s and 1960s, classical experiments indicated that isolated chloroplast can convert carbon dioxide to water to carbohydrate. And this photosynthesis, it involves two phases. One is the light reactions, in which oxygen is produced, ATP and NADPH are the products of the light reactions of the photosynthesis. And this carbon reactions, which reduces this carbon dioxide to carbohydrate. And for this reduction, it consumes ATP and NADPH. Actually, both this reaction occurs simultaneously. But this concept of water oxidation and this carbon dioxide reduction, they are not obligately linked. So this concept that light reactions and carbon reactions, that they are not obligately linked, that was advanced in 1930 when the Robert Hill showed that various non-physiological redox active compounds can act as the electron acceptor that is arising from the oxidation of the water. Actually, today I would like to concentrate on these carbon reactions. Actually, these carbon reactions in the past, that is the Calvin-Benson cycles, it was referred to as dark reaction of photosynthesis. But this terminology is misleading, because these reactions occur during the day. And many of the Calvin-Benson cycle enzymes, it requires light for their activation. So that means this carbon fixation reactions, it depends on the light reactions, both for high energy compounds like ATP and NADPH, as well as regulatory signal. And that regulatory signal, it regulates the flux through Calvin-Benson cycle. Now, if you consider the structure of the Rubisco, actually, before going to the protein structure, because Rubisco, it is an enzyme. So this enzyme is a protein, and protein is composed of amino acids. And actually, the protein structure can be visualized at 4 different levels. That is the primary structure, which defines the amino acid composition and sequence. And the secondary structure, the conformation without regard to the R groups, and tertiary structure. This tertiary structure, which is concerned with those protein which contains single polypeptide chain.
Sir, sorry for interruption. Sir, light is fixed, not change.
It is not changing?
No.
This tertiary structure, why it is so? Which is concerned with those protein which contain. But here it is changing. I don't know.
Sir, sorry for interruption. Sir, light is fixed, not change.
It is not changing at all.
Yes,
No,
sir.
first slide is visible only.
First slide.
Yes.
Okay, just this is not visible.
Only
This slide
first
this
slide
slide
is
is-
visible.
Eh?
First slide is visible only.
Okay, just wait a bit. Okay, then I am just again joining this. Now it is changing. Hello,
Yes, yes.
it is
Yes, sir,
changing.
changing. Yes, changing.
Then I will start from beginning, or okay, just so this. But earlier you have not seen these reactions. Okay, I think. Hello,
Yes.
you have seen this slide. What is in the book?
Yes, seen.
Next one, this one.
Yes, seen.
Okay. Then this one.
Seen.
Okay, then it's okay. So I was discussing with this one. Actually, this structure of the Rubisco. Actually, this Rubisco, it is the most predominant protein in plant leaves. Actually, this Rubisco, it catalyzes both the carboxylation and oxygenation of RUBP. And this Rubisco, actually this from the comparison of the primary structure of Rubisco, 4 different forms of Rubisco has been identified based on the composition of the large and small subunits. Because there are 2 types of subunits. One is the L, another is the large subunit, which has a molecular mass of 50 kD, whereas the small subunits is has a molecular mass of 15 kD. So these different forms, that is 4 different forms, that is form 1, form 2, form 3 and form 4, these 4 different forms differ in the arrangement of these large and small subunits. So these large subunits, you just see this Rubisco, that is the form 1, form 2 and form 3, they are catalytically competent. That means they can fix carbon dioxide. But the form 4, that is incapable of fixing carbon dioxide, they probably have different role in plant metabolism. But in all these different forms, the catalytic unit, it is composed of, it is homodimers. You just see, this is the homodimers, that is these 2 red squares, homodimers. So that means this 8 monomers, it forms the 4 dimers. Actually, this form 1, this is found in all autotrophic organisms, and it contributes 50% of the total protein of the stroma. And actually, its high level in photosynthesizing cells, as well as its presence in phytoplankton, provide more than 45% of the global annual net production of carbohydrate. So this form 1, this is the only form of Rubisco which harbors both the small subunit S4, and this is the small subunit, that is the S, and large subunit, that is the L. So these large subunits actually this form 1, this 4 large subunits, actually these large subunits, they form a complex which is capped by 4 small subunits from the top and 4 small subunits on the bottom. So this is the structure of the Rubisco, and this another forms, this is the form 1, 2, 3, 4. This form 1, that is the 8 large subunits and 8 small subunits, and the organisms, the plant, algae, proteobacteria, and cyanobacteria, they contains this form 1. And this is the only form which actually harbors the small subunit. And it is interesting to note that small subunit, it is encoded in the nucleus, but the large subunit, it is encoded in the chloroplast. But for the assembly, these nuclear encoded proteins must be imported to the chloroplast for the function of the Rubisco. Actually, if you see the this Rubisco activity, so this Rubisco, it has both carboxylation and oxygenation activity. And actually, you are familiar with this reaction, that's why I won't discuss this reaction. But what is important, that all Rubisco isolated thus far, even from obligate anaerobic prokaryotes, they retain the oxygenase activity. That means the oxygenase activity of Rubisco, that oxygenase activity, that is the intrinsic to Rubisco, and the ancestral enzyme was evolved in an anaerobic environment. But what is important, if you consider, that means the Rubisco, in addition to its carboxylase activity, it has oxygenase activity. And this oxygenase activity, that is the relative this both the carbon dioxide and oxygen, they compete for the same active site of the enzyme Rubisco. So this Rubisco activity towards carboxylation and oxygenation, it depends on the relative concentration of the carbon dioxide and oxygen in the atmosphere. Now, usually for land plants and the current atmospheric condition, the carboxylation reaction is 3 times faster than the oxygenation reaction. Actually, this ratio of carboxylation by oxygenation reaction, it defines the photosynthetic efficiency. So we can arrive at this ratio, that this velocity of carboxylation reaction by oxygenation reaction, it is 3:1, because if we calculate the velocity of carboxylation reaction Vc, that we can put the simple Michaelis-Menten equation where the Vmax into substrate concentration, Michaelis constant plus substrate concentration. Here, this Vmax, that is the capital Vc, signifies the Vmax of the carboxylation reaction. This is the carbon dioxide concentration, and Kc represents the Michaelis constant of carboxylation and plus carbon dioxide. Now, a simplifying assumption is made that carbon dioxide concentration is much lower than the kinetic constant, Michaelis constant for carboxylation reaction. So this reaction then reduces to velocity of carboxylation reaction equal to Vmax by Km, both for carboxylation reaction into carbon dioxide concentration. Similarly, velocity of oxygenation reaction can be written as V0, that is the Vmax of oxygenation reaction, and Ko means the Michaelis constant of the oxygenation reaction into oxygen concentration. So the ratio of Vc by Vo equal to this Vmax. This is a set of kinetic constant, that is Vc into K0 by Kc into V0 by carbon dioxide concentration by oxygen concentration. Now, this is called the specificity factor or selectivity factor. That means when the carbon dioxide concentration equals to oxygen concentration, then ratio of Vc by V0 equal to this set of kinetic constants, and that is called the specificity factor. For the most land plants, this specificity factor, it varies between 80 to 100, 130. So that's why if you assume this 100, it is the average value of specificity factor for the land plants, and this carbon dioxide concentration, which constitutes only 0.038%, that is the corresponds to 8 micromole, and the oxygen concentration, that is the 21%, that is 250 micromole. So this ratio comes to 3.2. So this ratio, actually this velocity, the rate of the ratio of carboxylation by oxygenation ratio, it defines the photosynthetic efficiency, and that can be expressed as their kinetic constants of the carboxylation and oxygenation reaction. Now, actually the enzymes which are regulated by light in the Calvin-Benson cycle, that is not only Rubisco, but also several other enzymes, that is fructose 1,6-bisphosphatase, where this 1,6-bisphosphate, it hydrolyzed to fructose 6-phosphate. Then pseudoheptulose 1,7-bisphosphatase, pseudoheptulose 1,7-bisphosphate is converted into pseudoheptulose 7-phosphate, and this phosphoribonuclease, that is the penultimate step of the regeneration reaction, where 3-ribulose 5-phosphate is converted into ribulose bisphosphate, and another is the NADP glyceraldehyde 3-phosphate dehydrogenase. So this gap is reduced to glyceraldehyde 3-phosphate is reduced to 3-phosphoglycerate. Okay. So now this actually why the light regulation is required for the regulation of this Calvin-Benson cycle. Actually, this light regulates the flux through Calvin-Benson cycle. And this light regulation is critical. Why it is critical? Because this chloroplast stroma, it contains both Calvin-Benson cycle enzymes that catalyzes the synthesis of carbohydrate. At the same time, chloroplast stroma, it contains the enzymes of oxidative pathway which catalyzes the degradation of carbohydrates. So we can say if both the process, this Calvin-Benson cycle that catalyzes the synthesis of carbohydrate and the enzymes of oxidative pathway, both this pathway operates at the same time, it will be a futile cycle. So in order to prevent cycling, futile cycling, and the ensure optimal activity, it is essential for the synthetic apparatus to be on and the degradative apparatus to be off in the light. So this is that is if in the light the synthetic apparatus is on, certainly the degradative apparatus must be off. Actually, this any enzymes, its activity is controlled at two levels. Actually, the level of the enzyme is important, and another is the catalytic activity of the enzyme is also important. Actually, the chloroplast, it ensures appropriate rates of biochemical transformation through modification in the level of enzymes and the catalytic activity of the enzymes. So now if you consider this, actually in the light, actually the specific regulatory mechanism is there, and this specific regulatory mechanism, it mediates it activates the Calvin-Benson cycle enzymes when needed in the light and turn them off in the dark. On the other hand, the enzymes of the oxidative pathway, particularly pentose phosphate pathway, because you just see both this process, pentose phosphate pathway, it is the major source of NADPH. And this photosynthesis, light reactions, it is also a major source of NADPH. So this specific regulatory mechanism, it activates the enzymes of the Calvin-Benson cycle when needed in the light and turn them off in the dark. And whereas the enzymes of the oxidative pathway, particularly pentose phosphate pathway, and more particular specifically glucose 6-phosphate dehydrogenase reaction, it operates in opposite manner. That means it is inactive in the light and active in the dark. So actually we are talking about the rates of biochemical transformation. It is controlled by the level of enzymes. Actually, these stromal enzymes, the sum of the enzymes, they are encoded in the nucleus, and some of the enzymes, they are encoded in the chloroplast. That is the Calvin-Benson cycle enzymes. They are encoded in both nucleus and chloroplast, and levels of these enzymes depend on their rates of gene expression and protein synthesis. For example, this nuclear encoded genes are translated on cytosolic ribosomes, and these proteins have a N-terminal peptide signal, and that peptide signal, it is cleaved at the inner membrane of the chloroplast envelope, and as this mature enzyme enters into the chloroplast and ultimately released into the stroma. Likewise, the large subunits, they are actually this protein synthesis. This chloroplast encoded proteins are translated in the prokaryotic like plastidic ribosomes. So to build the actual amount of enzymes for the stroma, actually the expression of the nuclear encoded gene must be coordinated with this chloroplast encoded genes. So actually this regulation, this coordination, actually upon illumination, actually this when the light stimulates the expression in such a way that synthesis of the both chloroplast and nuclear mRNA is coordinated, and it ensures that both the proteins are present together in sufficient amounts to carry the activity. Now, this is the interaction, that is the signal transduction between the nucleus and the chloroplast. It may be anterograde or retrograde. Where this anterograde, the products of the nuclear gene controls the transcription and translation of the plastid genes. You just see the small subunits of Rubisco, they are encoded in the nucleus. So the precursor of this nuclear encoded products, until they are present in sufficient amounts in the stroma, they cannot lead to the translation of the plastid genes. On the other hand, in some cases, this signal transduction is retrograde. That means the signals flow from the chloroplast to the nucleus. For example, this ferrochelatase. This is an enzyme which is responsible for the hemisynthesis in the plastid, and this ferrochelatase actually it triggers the translation of the nuclear genes. Now let us consider actually this accumulation. If the protein is formed after post-translation, after translation, but the simply accumulation of the enzymes, it causes slow changes in the activity. Actually, after the formation of the protein, this protein undergo post-translational modification to change the activity in response to changes in the redox condition of the stroma. I shall discuss later how the redox condition of the stroma is changed. Okay, so because of the change in redox condition of the stroma, there is a post-translational modification of some of the enzymes, particularly fructose bisphosphatase, this pseudoheptulose 1,7 bisphosphatase, phosphoribullokinase, and the NADP-GAPD. So actually, this Calvin-Benson cycle enzymes, they actually acquire catalytic activity by two mechanisms, and both these mechanisms are associated with the alteration of the protein structure. So first one is the Rubisco activase, another is the Ferredoxin Thioredoxin system. So this Rubisco activase, it activates Rubisco, whereas this Ferredoxin Thioredoxin system, it activates fructose bisphosphatase, pseudoheptulose bisphosphatase, phosphoribonuclease and NADP-GAPD. Actually, the unique mechanism that regulates Rubisco activity that is related to the slow capacity of the Rubisco to fix carbon dioxide. Rubisco, it has very slow capacity to fix carbon dioxide, that is 1 to 12 carbon dioxide molecules fixed per second per active site. So this is very slow. That's why this prior to catalysis, this Rubisco is activated by the carbon dioxide. But you just see this carbon dioxide, it is a substrate of Rubisco. At the same time, it is a modulator of the Rubisco. So how is the Rubisco is modulated by carbon dioxide? So this Rubisco, this is a protein. Protein is composed of amino acids. So at the 243rd position of the Rubisco, there is a lysine residue, and this lysine residue contains the epsilon amino group. So this is the Rubisco enzyme. At a specific position, there is a epsilon amino group. So this is a part of the lysine residue, and here is the carbon dioxide. So when this electron moves towards the carboxyl oxygen by electromagnetic effect, so the unshared electron pair of nitrogen, it attacks this carbon. So in this way, a carbamoyl derivative is formed. This is a carbamoyl derivative, carbamate is formed. Now here, this oxygen, it has unshared electron pair, and oxygen, it is negatively charged. Actually, this magnesium shields these negative charges. That's why this prior to catalysis, like the Rubisco initially, it has very low capacity to fix carbon dioxide. That's why this prior to catalysis, Rubisco is activated by carbon dioxide. So this carbon dioxide reacts with the specific lysine residue, epsilon amino group of lysine residue, to form the carbamate derivative. And this carbamate derivative then combines with the magnesium to form a ternary complex. Therefore, this is up to an age, this is the part of the enzyme. So this carbon dioxide, this is the another component, and this is the magnesium. So this is ternary complex. This is the catalytically active enzyme. So this ternary complex, actually, it binds the RUBP. Here it is not shown. This is this carbon dioxide, this ternary complex. Here this RUBP is bound. So this is the complex. Then carbon dioxide reacts with this RUBP to give two molecules of 3-phosphoglycerate, and when it reacts with this oxygen, it gives 3-phosphoglycerate and 2-phosphoglycolate. So that means here you just see this is the epsilon amino group of lysine residue of the Rubisco. Then proton is removed and carbon dioxide enters. It forms the carbamoyl derivative. Then magnesium binds. Then it forms this ternary complex. So this ternary complex, that is the two substrates of this Rubisco. One is the carbon dioxide, another is the RUBP. So this carbon dioxide, it reacts with this RUBP to form two molecules of 3-phosphoglycerate. Okay. So now another interesting fact. This is the carbamylated Rubisco. You just see this ternary comment. This is the carbamylated Rubisco, and this is the uncarbamylated Rubisco. And both this carbamylated Rubisco and the uncarbamylated Rubisco, they are subject to inhibition by the natural sugar phosphate. So this natural sugar phosphate, because if you see the reaction mechanism of the carboxylation reaction, because the C5 intermediate, that is the RUBP, And when carbon dioxide is added, then that is the C6 intermediate. So this C6 intermediate, there is the natural sugar phosphate, that is the this is the intermediate of the Rubisco reaction, that is the 2-carboxy 3-keto-arabidiol 1,5-bisphosphate, and this is a natural sugar phosphate, that is 2-carboxy arabinotol 1-phosphate. So actually this sugar phosphate, it can bind to both carbamylated and uncarbamylated Rubisco. That's why it has been shown that in many plants, what happens, the Rubisco activity is low in dark adapted leaves, and that Rubisco activity cannot be reactivated by the addition of carbon dioxide and magnesium, because I have already mentioned this carbon dioxide, it actually activates Rubisco, forming the carbamyl derivative, and this followed by magnesium binding. And Rubisco activity in leaves, in dark adapted leaves, it is very low, but that leaves cannot be, this Rubisco activity cannot be reactivated by the addition of carbon dioxide and magnesium. So this is one thing. And another thing, if you collect the Rubisco from the same leaf during the day, that can be activated by this carbon dioxide and make magnesium. Actually, in support of this experiment, what happens, this uncarbamylated Rubisco, and here this is the sugar phosphate. So this sugar phosphate binds with this uncarbamylated Rubisco to forms the Rubisco and sugar phosphate. Now this Rubisco activates, this is for the activation of this Rubisco activates, it requires ATP. So this Rubisco activates, it removes the sugar phosphate from this inactive Rubisco. So ultimately it comes to this pool. Actually, this why this ATP is required for this activation of this Rubisco activates, it is not yet understood. But this is the case of the uncarbamylated Rubisco. But in case of the carbamylated, because you just see, this is the carbamylated Rubisco, this is the active. So this is the carboxy arabinotol 1-phosphate. So it combines with this. Okay. So this Rubisco activates, then removes this Ca1P, and in the light, this phosphatase removes this phosphate group, it gives the Ca. Then this Ca is in the dark by phosphorylation at the expense of ATP, it is converted into Ca1P. So that's why, because in the light, actually this activation of the Rubisco in the light, it involves the Rubisco activase and light dependent degradation of this sugar carboxy arabinotol 1-phosphate to Ca. So that's why in many plants, the Rubisco activity is low in dark adapted leaves, and they cannot be activated by treating carbon dioxide and magnesium. But the same Rubisco, if it is obtained during the day, and that can be reactivated by this treatment. So there is these two mechanisms. In the light, the Rubisco activates, it removes the sugar phosphate from this inactive complexes of the Rubisco, and in the light, there is a light dependent degradation of this sugar phosphate by the enzyme phosphatase. So now this Rubisco activase in many plants, this Rubisco activase, it occurs in two isoforms. One is the longer isoform, another is the shorter isoform. This longer isoform, it has the molecular weight of 47 kD, whereas the shorter isoform, it has molecular weight of 42 kd. Actually, both these forms arises for the alternative splicing of this mRNA. Actually, this longer isoform, it contains an extra peptide at the C-terminal region and which has two cysteine residues. So these two cysteine residues, this is the cysteine, it is the among the 20 amino acids, it is a very unique amino acids because it can reversibly form the disulfide linkage by oxidation, and by reduction it gives the thiol or sulfhydryl groups. So that means this extra peptide at the C-terminal region with two cysteine residues, actually these cysteine residues, they can undergo this reversible change between this disulfide linkage and sulfhydryl group. So what happens, actually these longer actually plants that express this longer isoform, the photosynthesis is inhibited in the longer isoform, that plant that expresses this longer isoform due to heat stress. But the plants that express this shorter isoform, their photosynthesis is not affected. Okay, so that's why the studies with Arabidopsis mutants indicates that photosynthesis in plants that expresses the longer isoform is inhibited due to heat stress than the plants that expresses the shorter isoform. But whether this Rubisco activates, certainly they activate Rubisco, but whether this Rubisco activates this longer isoform and this shorter isoform, their role is associated with the heat stress resistance, that is not yet clearly known. It is only known this longer isoform, their photosynthesis is adversely affected than the plants that expresses the shorter isoform. So that means this shorter isoform, it has some role in heat stress, but this has not yet convincingly established. Now, another interesting fact is that the second is that this is all about the regulation of the Rubisco and these other mechanisms by which these other enzymes of the Calvin-Benson cycle are regulated. So what happens, during the day, the light reactions, it links the oxidation of the water to the reduction of the NADP to NADPH. Or you can say it can links the oxidation of the water, links the reduction of the Ferredoxin to reduction of the Ferredoxin. So during the day, this electron transfer, light-mediated electron transfer, it generates the intermediates.
Sir, please select slide slow.
Select
Slide show.
Achha. It is not actually visible.
Visible, but very short in figure.
Okay, just. Okay, now.
Sir, please change slideshow, sir. If you change it slideshow, then it shows in large.
Now it is large.
No, sir.
But I have given it a slide, so.
Oh, yeah. Then continue, sir. Continue.
Continue,
Sir. Okay, sir. Continue, sir.
So what happens during the day? This light-mediated electron transfers, it generates the intermediates. So what are these intermediates? This may be reduced Ferredoxin, or the NADPH. The interesting property of these intermediates that they have high negative reduction potential. If the reduction potential of a couple, because if you couple means ferredoxin, it can alternate between the oxidized and reduced state. So in this couple, this electron transfer affinity, it is given by the redox potential. If the redox potential is negative, that means it has low potential, low affinity for the electron. But if this redox potential has a positive value, that means it has high affinity for electron. So this light mediate electron transfer, it generates intermediates which have high reducing potential. For example, this ferredoxin, ox to ferredoxin red, this couple has redox potential of minus 420 millivolt, whereas the NADP to NADPH couple, this couple has redox potential of minus 315 millivolt. That means this negative value signifies that these intermediates can readily transfer their electron to another species. So this light mediate electron transfer generates intermediates with high reducing potential, and this high reducing potential can transform this oxidized inorganic precursor to the organic, reduced organic metabolite. So what is important here, accompanying this electron transfer, a network of redox processes, you just see here the water is oxidized in the luminal side, then electrons moves from the cytochrome B6 complex in pH1, then ferredoxin. This network of redox processes, it transfers information to the chloroplast stroma, because water is oxidized in the lumen. The electrons are carried across this thylakoid membrane, and ultimately these electrons are transferred to the ferredoxin. Accompanying this electron transfer, a network of redox pathways transfer information from the thylakoid membrane to the chloroplast stroma. And when this information is transferred to the chloroplast stroma, then it activates the several stromal enzymes which are required to carry out this Calvin-Benson cycle. So what is important here, actually you have seen, I have already discussed that cysteine, this is the unique amino acids among the 20 amino acids. So in the cysteine amino acid, in the side chain, there is a sulfhydryl group. So these cysteine sulfhydryl groups can form the disulfide linkage by oxidation. Similarly, this disulfide linkage by reduction, it can form the sulfhydryl group or thiol linkage. This ability of the cysteine residues to form intramolecular or intermolecular reversible disulfide linkage and thiol form, that is of primary significance for the dynamic regulation of the stromal enzyme in response to the reducing condition of the stroma. That means what we can say, in the light, the stromal condition is reduced. In the dark, the stromal condition, redox state is the oxidized state. So this ability of the cysteine residues to reversibly form intramolecular and intermolecular disulfide linkage, that is important for the dynamic regulation of certain Calvin-Benson cycle enzymes in response to the redox condition of the chloroplast stroma, and the enzymes which are actually regulated by this mechanism, that is the fructose bisphosphatase, pseudoheptulose 7-phosphatase, and this phosphoribullokinase, and NADP GFP. So these are the 4 enzymes which are regulated. Actually, this post-translational modification, protein, when it is by translation, the protein is formed in that protein, cysteine residues, they are present in the disulfide linkage. But when this network of redox pathways transport information from thylakoid membrane to the stroma, that this disulfide linkage is converted into the sulfhydryl group or thiol group. That means we can say this actually post-translational modification in which this disulfide linkage is converted into the sulfhydryl group. This has been the subject of intense research since 1970, when this Ferredoxin Thioredoxin cysteine was discovered. So what happens actually, this during photosynthesis, the electrons, this electron flow from this Ferredoxin, because we just see this electron, this photosystem 2 and photosystem 1, that catalyzes the oxidation of the water to oxygen. The electrons ultimately goes to Ferredoxin and it gives the reduced Ferredoxin. Actually, during photosynthesis, this electron flow from the reduced Ferredoxin, which is generated by the light reactions, to the target enzyme. So this is the target enzyme. So this target enzyme is the fructose bisphosphatase, pseudoheptulose 7-phosphatase, phosphoribullokinase, and the NADP-GAPD. So these are the 4 enzymes. So during photosynthesis, this electron flow from the reduced Ferredoxin to the target enzyme. But this electron transfer is not direct to this. Rather, this involves 3 proteins. One is the Ferredoxin. It is a non-hemi iron sulfur protein. Okay, or it is, you can say, it contains the iron sulfur cluster 2Fe2S, and that is also a iron sulfur protein, FTR, Ferredoxin Thioredoxin reductase, and here is the Thioredoxin. So in all these proteins, you just say there is the disulfide linkage, and that disulfide linkage, it alternates between this disulfide, oxidized disulfide, and reduced thiol. So what happens? These 3 proteins actually form a complex. There is Ferredoxin, FTR, and Thioredoxin, and these 3 proteins form a complex. Okay, so this complex converts this reducing power here, this reducing power, to the regulatory thiol signal in the target enzyme. This reducing power, it is originated by the light reactions of the photosynthesis, and in the thylakoid membrane, this reaction, and ultimately it transferred to the chloroplast stroma for activation of this target enzymes. So what happens? This redox transformation begins when this reduced Ferredoxin transfers electron to the Ferredoxin Thioredoxin reductase and cleaves this disulfide bond to this sulfhydryl form. So this reduced Ferredoxin reductase then transfers this electron to this Thioredoxin, and this disulfide linkage is cleaved to reduced sulfhydryl level, okay, reduced Thioredoxin. And once this reduced Thioredoxin is formed, this redox transformation is complete. When this reduced Thioredoxin, it transfers this electron to the target enzyme, and it converts to this sulfhydryl form, and thereby activate the enzyme. And this electron diffraction studies, X-ray diffraction studies actually indicate this complex, that ferredoxin, FTR, and thioredoxin, they form a complex. Actually, this ferredoxin, that is the electron donor, and this thioredoxin, that is the electron acceptor. Both these proteins, they are docked on the opposite sides of the ferredoxin, thioredoxin, or the docking of the ferredoxin is opposite to the docking of this thioredoxin. So in this way, this information, this network of redox pathways, it is transferred from thylakoid membrane to the chloroplast stroma for the regulation of certain Calvin-Benson cycle enzymes. Now, how then oxidized form, that the reduced form is the active. So this thioredoxin reduced, that is the mechanism by which this thioredoxin reduced thioredoxin form, that is, it is reduced by thioredoxin, this enzyme. So this is the inactive form and the active form. So this how they converted from the active to inactive form in the dark, it is not clear. But what is known, actually in the dark, this electron flow from this thioredoxin, it ceases. And another, then here lies the mystery, that these oxidative enzymes of the pentose phosphate pathway, they are inactivated in the light. So what happens actually in this case, you just see the ferredoxin, it is the light react product, ferredoxin ox is converted into ferredoxin reduced. So similar to, so here, this is the light reaction, this NADPH can be produced, and that can be reduced to reduce carbon dioxide to carbohydrate. And here is the pentose phosphate pathway, glucose 6-phosphate dehydrogenase, that also gives 6-phosphogluconolactone. Okay, so this phosphoglucone, this NADPH is converted into NADP. If the light reaction is active, the degradative pathway must be inactive. So what happens? So when this in the light, this ferredoxin ox is converted into ferredoxin red. So this electron, it preferentially go to reduce NADP to NADPH. But at the same time, this part of the electron, it moves to this ferredoxin thioredoxin system. Therefore, this ferredoxin, it goes to the thioredoxin, and ultimately this thioredoxin, this disulfide linkage of the glucose 6-phosphate dehydrogenase, it is converted. So it is active when this disulfide linkage is there, but it is inactive when these cysteine residues are present in the reduced form. So here, when these electrons, it moves in the both the directions. So actually, this glucose 6-phosphate dehydrogenase becomes inactive, and this becomes active. And that's why this in the dark, actually, when the plants require to reduce nitrate, sulfate in addition to carbon dioxide. So in that case, there is a great demand of this NADPH. So in that case, the electrons, they are preferentially transferred to this glucose 6-phosphate dehydrogenase in the dark. Okay, so in this way, they are regulated. So the next one, that is also very important. So you have seen these 4 enzymes, they are regulated by ferredoxin, thioredoxin system. But another mechanism is Egra, because in the nature you will see there is a canopy cover or cloud cover. That means there is a dark to light transition. So what happened? The photosynthesis is also, this Calvin-Benson cycle enzymes are also regulated by this dark to light transition. So these proteins, this Calvin-Benson cycle enzymes, they undergo non-covalent interactions with some other proteins. That is protein-protein interactions. Because if you consider PRK, this is a protein as well as enzyme. So it interacts with the other protein. That the means of interactions, that is the non-covalent interactions. So these non-covalent interactions, it may include hydrogen bonding, electrostatic forces, hydrogen bonding, or hydrophobic interactions. So these non-covalent interactions of the many enzymes of the Calvin-Benson cycle with other proteins as well as metabolites like ATP and NADPH, these are the products of the light reactions. So these interactions can alter the catalytic activity within minutes of dark to light transition. So what happens here, you just see these are the three proteins. So what happened, this is the assembly of three proteins. So this assembly of the non-covalently linked complexes, it causes changes in the activity to subtle changes in the intensity and quality of light. And this intensity and quality of light, that is actually observed during cloud cover or canopy cover. So that these proteins, so suppose this is one protein, so this is another protein, and this is another protein. I mean, these three proteins, they undergo non-covalent interactions, and these non-covalent interactions.
Sir, sorry to disturb, sir. Sir, change the slide, sir.
Change the slide.
In slide mode, to give it to slide mode, then it will be bigger.
Figure
Slideshow
is
mode.
not changed. Figure is not changed.
Okay, just
Please change the figure.
okay, just it is changing. Okay,
Yes, sir.
No.
Okay, this is why I was showing this one. Now it is changing. Okay, it has changed.
No.
But here it is changing, but I don't know what is wrong with it.
No. But skin showing the steel, mammalian organization of jet skin only.
Okay, just
Yes, change.
same.
Yes, changing. Yes, sir.
Changing.
Yes.
Okay.
अरे फोन नंबरটা দিয়ে গেলেন না
Okay.
Okay, sir.
Actually,
Please start, sir.
so actually this many Calvin-Benson cycle enzymes, they undergo this non-covalent reactions with other proteins as well as metabolites like ATP and NADPH. And these non-covalent interactions can alter the catalytic activity within minutes in dark to light transition. Actually, this assembly of non-covalently linked complexes, they cause rapid changes in the activity of the associated enzymes to subtle changes in the intensity and quality of light. So this intensity and quality of light, that varies according to this cloud cover and canopy cover. Actually, many of these Calvin-Benson cycle enzymes, they actually interact non-covalently with some regulatory proteins, and it forms the supramolecular complexes like this. So they can also interact with the metabolites of the stroma like ATP and NADPH, they can also respond to the pH and ionic composition of the stromal region, and they can also interact with some proteins of the thylakoid membrane. So these non-covalent interactions, they actually act in concert with the Ferredoxin-Thioredoxin system. It is not a separate. So these interactions, non-covalent interactions, they act in concert with Ferredoxin-Thioredoxin system to maximize the catalytic activity of the Calvin-Benson cycle enzymes. So you have seen earlier this PRK, phosphoribonucleokinase, and NADP-GAPD, these two enzymes are regulated by Ferredoxin-Thioredoxin reduced system. But in addition to be reduced by Ferredoxin-Thioredoxin system, these two proteins, they also form non-covalent interactions with CP12. So this is the CP12 protein. So this CP12 protein, this is a chloroplast protein, but it is encoded in the nucleus. So this is CP12, this is the regulatory protein. So this PRK and this PRK, it is a homotetramer. This is NADP-GAPD, or that NADP dependent glyceraldehyde 3-phosphate dehydrogen. It is a homotetramer. There are 4 subunits. So this CP12, this is actually CP12. It is a chloroplast protein. It is encoded in the nucleus, and it contains 4 cysteine residues, 1, 2, 3, 4. And this is the PRK. This is a dimer. This is one subunit. This is another subunit with 4 cysteine residues. Actually, this PRK and this NADP-GAPD, these two proteins, in addition to being regulated by Ferredoxin-Thioredoxin system, they form non-covalent interactions with regulatory protein CP12. So in the light, that means the stroma is in the reducing condition. So in the light, that is in the reducing condition, most land plants contains the active NADP-GAPD, and they contain, that is a homotetramer, and contains the active PRK that harbors 4 cysteine residues, and it contains the CP12 in the reduced condition, that is 4 cysteine residues which are in the reduced condition. So what happens in the dark? So this sulfhydryl group, they are oxidized. So these sulfhydryl groups, they are oxidized of CP12 and this PRK. So these oxidized form of both PRK and CP12, it recruit this NADP-GAPD to form this supramolecular complex by non-covalent interactions. So in the dark, actually, these disulfhydryl groups are oxidized. So these oxidized CP12 and PRK12, they are recruited. They recruit this NADP-GAPD to form the supramolecular complex. And within this complex, both these enzymes, that is PRK and this NADP-GAPD, they exhibit marginal activity. So when in the light, this Ferredoxin-Thioredoxin system, that is activated. So that reduces this disulfide linkage. Okay, so this disulfide linkage, ultimately the associated proteins, they are released from this supramolecular complex and they exhibit their full catalytic activity. But interestingly, there is a considerable heterogeneity, because in dark, it can form this CP12 it can form this assembly, non-covalently linked assembly in the dark adapted B. But when the CP12 in tobacco is suppressed by genetic manipulation, very minor changes in the photosynthetic rate and the activity of these two enzymes. That means whatever is this P, these they form in dark adapted chloroplast. In P, it forms this non-covalently linked complexes. But when this CP12 is suppressed by genetic manipulation, it causes minor changes in the activity of these two enzymes. But nevertheless, it leads to a change in the growth rate of the plant and the carbohydrate partitioning and the morphology. That means this assembly, it is formed in the P, but it is not formed in the tobacco. That means there is a considerable heterogeneity. So this considerable heterogeneity indicates that this CP12, the role of CP12, it extends the role of this non-covalent interactions with these other proteins. And now it has been observed in many plants, this NADP GAPD, you have seen it is a homotetramer. But there are some land plants which is this NADP GAPD, that is a heterotetramer. That means it contains two types of subunits. So you just say this is the A subunit and another is the B subunit. So that's why it is the heterotetramer. So this is heterotetramer, that is A2B2. Heterotetramer, actually this B subunit, that is very homologous to the A subunit, but it has an extra peptide in the C terminal side with 2 cysteine residue in each B subunit. If this gray color is the B subunit, so this B subunit is homologous to the A subunit, and this B subunit contains an extra peptide with 2 cysteine residues. So what happens in the light, that is the reducing condition, that is Ferredoxin system, when it is activated, so this sulfhydryl group, this cysteine residues, they are in the sulfhydryl form. But in the dark, it is converted. Actually, the sulfhydryl groups are oxidized. So this oxidized form, they undergo polymerization to form a hexadecameric structure, A8B8. So in this way, it inactivates the NADP GAPD. So in this way, these two enzymes, this NADP, it is also activated. Now, what is important, this magnesium, if you see in this slide, when this dark adapted chloroplast, it is exposed to light, the stromal pH increases. This small side, the stromal pH increases because the protons moves from stroma to the luminal side. Okay, this light drives the translocation of the proton from the stromal region to the luminal side. Now, accompanying this proton transfer, actually to compensate for the influx of the positive charge, the magnesium is released from the lumen. So it enters into the stroma. So as a consequence, the stromal pH, it increases from 7 to 8, and the magnesium concentration, it increases from 1 to 3 millimole to 3 to 6 millimole. So this change in stromal pH and magnesium concentration, it complements the Ferredoxin Thioredoxin system, as well as it activates some of the enzymes of the Calvin-Benson cycle, like fructose 6-phosphatase, pseudoheptulose bisphosphatase, and Rubisco. And finally, I would like to mention with respect to this, actually this, now we are very much worried about this global climate change and photosynthesis. So we are mainly concerned with the C3 plants. So what happens here, earlier I have shown you this equation that describes the photosynthetic efficiency. This is the equation. So the equation that describes the photosynthetic efficiency, that is the ratio of carboxylation and carboxygenation reaction. But what has been observed, that if global climate change, that is temperature, global warming, it is an issue. So what we can predict. So this, what has this quantum yield, actually the per quanta absorbed, the net carbon dioxide fixed per quantum absorbed. So in case of C3 plant with increasing temperature, this actually decreases. And it has been shown this optimum temperature, it is nearly about 25 to 30 degrees centigrade in C3 plant. So what is the reason? Because in case of C3 plant, which has experienced both carboxylation and oxidation activity, and this oxygenase activity that ultimately leads to the photorespiration. So in that case, this C3 plant with increasing temperature, the net carbon dioxide fixed per quanta absorbed, it decreases with time, with temperature. Why this? And it has optimum temperature nearly 25 to 30 degree centigrade. Now, happens here? This is the equation which describes the photosynthetic efficiency, Vc by V0. Okay, so this set of kinetic constants is called the specificity factor. So this specificity factor, when we considered, it is 100, and the carbon dioxide concentration is the 8 micromole, and oxygen concentration is the 250 micromole. So this ratio becomes 3.2. So if there is a rise in temperature, so this will change the kinetic constants of the Rubisco. So ultimately, this decreases the specificity factor of the Rubisco. If earlier it was 100, if it is reduced to 80, so certainly decreases the carbon dioxide capture. And another consequence, if the temperature increases, so there is a decrease in the solubility of the dissolved carbon dioxide, much more than that of oxygen. Now, this available carbon dioxide by available oxygen, this ratio, it decreases. So these are the two important effects that is observed in high temperature. Actually, these two effects ultimately associated in the decrease in the carbon dioxide capture. The first one, this temperature increase, it increases change the kinetic constants of Rubisco and decreases the specificity factor, okay, and decreases the carbon dioxide capture. And the second consequence, it decreases the solubility, because both the carbon dioxide and oxygen, they compete for the same active site. So this relative activity of Rubisco towards carbon dioxide or oxygen, it depends on the relative concentration of the carbon dioxide and oxygen. Like if temperature rise decreases the solubility of the dissolved carbon dioxide much more than that of oxygen, so available carbon dioxide concentration decreases. So automatically there is a decrease in carbon dioxide capture. Now another issue, that is there is a possibility, the projected doubling of the carbon dioxide concentration sometimes in this century. So what would be this consequence in this C3 plant? Because certainly if this projected doubling, this projected doubling of the carbon dioxide concentration in this atmosphere certainly have influence, because if this carbon dioxide concentration earlier we considered 8 micromole, so now it is doubled, that is 16 micromole. So this ratio becomes more than 6. So that's why there is a theoretical prediction. We can say that theoretically, if the carbon dioxide concentration in the atmosphere increases, the ratio, which is under normal atmosphere it is 3, it will be approaches to greater than 10. So that means it is advantageous for this C3 plants. But in reality, this is not the case, because although this carbon dioxide trapping efficiency increases with greater carbon dioxide concentration, but the net photosynthetic rate may not be. So actually, it has been numerous studies indicate that the plants, many plants acclimate to exposure of high carbon dioxide concentration such that their photosynthetic rate under high carbon dioxide concentration is similar to that of plants grown under normal atmosphere. So what is the reason behind this response? Theoretically, we can predict, but in reality, this is not observed. So they have, actually the plants, they acclimate to exposure to high concentration of carbon dioxide such that the photosynthetic efficiency, it is more or less similar to the plants that is grown under normal current atmosphere. So the reasons for this response that because diminished rate of Rubisco, because any enzyme activity, it depends on the two factors. One is the level of the enzyme is important, another is the catalytic activation of the enzyme. So that's why carbon dioxide concentration may be high, but the level of Rubisco is diminished at the high carbon dioxide concentration. That's why they are diminished level of Rubisco, they are unable to fix this enhanced carbon dioxide. And another is the feedback inhibition. That is, it is the inability to utilize the additional carbohydrate formed under excess carbon dioxide concentration. If there is, suppose Rubisco level is okay, the carbon dioxide is fixed, so the additional carbohydrate that is formed, that can inhibit the Rubisco. So these are the possible reasons. So whatever, regardless of the acclimation effect, actually this increase in temperature have impact on the balance of this photosynthetic and photorespiration reactions with unknown consequence. That is, still we don't know what would be the consequence. Okay, thank you all.
Thank you, sir,
Okay.
for your valuable and very informative lecture for our participant as well as our students. So, as the portion is under our syllabus of Vidyasagar University, so students should be more enriched, and as the lecture would be enriched then and to be benefited also. Now, I inform to the participant, if you have any question, you can directly interact to our resource person, Professor Pal. Or you may write any question in your chat box, or may directly interact. If you have any question, directly you can interact.
Sir, I have a question. Hello.
Yes.
Hello.
Yes. Proceed.
Sir, I have a question. Why Rubisco does not create ROS protein?
What? Pardon?
Why Rubisco does not create ROS protein?
It is not clearly audible to me actually.
Okay, sir. Repeat. Repeat it.
Once more.
Hello. Why Rubisco does not create ROS protein?
ROS protein.
ROS.
Yes. How do they produce ROS proteins, Rubisco? Because there is a possibility, Rubisco not like this, because in the last lecture I have done this, Dr. Nandi was telling, this ROS, this NADPH oxidase. Actually, this particular, there is a link between the ROS generation and the photosynthesis. But how it is linked? Because nowadays, if the optimum growing condition is there, this ROS generation, it must be balanced with the plant's inherent defense system. Because this generation of the ROS is inevitable in the plant. You cannot control. But at the same time, the plants, they have a well-defined machinery, both the antioxidative of enzymatic in nature and non-enzymatic in nature. They can scavenge this ROS. That's why they are in balance. But when this balance is disrupted, so in that cases, the ROS will be generated at an elevated rate. But I think there is a link up between the photosynthesis and this ROS generation. To me, it seems, because when plants are growing under optimum growing conditions, then it's okay. But when the plants are growing under adverse conditions, suppose the light energy is in excess, the stomata closer opening is such that there is a limited entry of carbon dioxide. So when the light energy is excess and carbon dioxide concentration inside the plant, that is the limiting, what would be the fate of the excess NADPH that is formed by the light reactions of the photosynthesis? So in that case, this NADPH oxidase, it will produce the superoxide radical by one electron, accepting one electron. Then 2 electron acceptor, it will give the hydrogen peroxide, and 3 electron acceptor, it will give the hydroxyl radical. So all are partly reduced oxygen intermediates, and they can affect the biological membranes. This is it. So what happens in that case? Actually, the plant, there is actually machinery, the excess NADPH when carbon dioxide is limiting. So just to prevent the ROS generation, this NADPH is diverted to the production of the secondary compounds like phenolics. So this is considered as plant, this phenolics, this is an energy escape valve. And that light energy is sufficient, but the carbon dioxide is limiting. So this, to prevent the oxygen, this ROS generation, ultimately this NADPH, they are diverted to the synthesis of the phenolic compounds. So in this way, the phenolics, they act as energy escape valve. Okay, what is it? Because this ROS, Rubisco cannot generate as such ROS, because this ROS generation, that there are several sites. Because in aerobic metabolism, if you consider the respiratory pathway or this photosynthetic pathway, there is the oxygen involved. So oxygen is the terminal electron acceptor. So this 4 electron reduction of oxygen molecule, it will always give the safe product. But when oxygen undergoes 1 electron reduction, it gives superoxide. 2 electron reduction, it gives hydrogen peroxide. 3-electron reduction, it gives hydroxyl reduction. So all are partly reduced intermediates. So these all partly reduced in a like oxygen, they are also oxidizing agent. But if you consider the hydroxyl radical, it is the more powerful oxidizing agent, because the oxygen to water, this couple has redox potential of plus 0.815 volt. But if you consider hydroxyl radical to water, this potential has 2.13 volt. That means hydroxyl radical, it is 3 times more powerful oxidizing agent than oxygen. So that's why if oxygen participate in any process, then in aerobic metabolism, there is inevitably there will be formation of the ROS. But this plant at the same time, they have a well-defined machinery, that is the antioxidant machinery, consisting of superoxide dismutase, consisting of catalase or glutathione reductase, or you can say other enzymes, the other non-enzymatics, because this phenolics, they are also non-enzymatically scavenge this radical species, or the glutathione, this is a tripeptide, so that can also act non-enzymatically. So there is always a balance between the generation and the scavenging. So they are always in balance, and when there is a disruption in the balance, so this ROS generation, it will form at an elevated rate, and it will give the adverse effect, because all these membranes, they are composed of the lipids. There is the unsaturation. So these radical species, they will attack this double bond, and the membrane is disrupted. If this membrane is disrupted, the contents within the membrane, they are leakage, and they are also susceptible to degradation by this ROS. Okay.
Thank you, sir. So I have another question. Why Rubisco function in particular molecular weight into dark reaction?
What?
Why Rubisco function in particular molecular weight into dark reaction?
Particular molecular
weight.
What does it mean?
Particular molecular weight into dark reaction.
Mane Rubisco of particular molecular weight.
Yes.
And actually, I think if it acts in the dark, that is this. If you consider, I don't know about this, how the molecular weight of the protein is modified, because it is the Rubisco is always composed of this plant Rubisco, that is the form 1. It is composed of 8 large subunits and 8 small subunits. That is the L8, S8, the stoichiometry of the Rubisco. But how this molecular weight of change, that is not understood. I don't understand this point actually. Certainly, this is universal. This molecular weight, this cannot be changed.
Okay.
I think.
Sir, I have another question. Why Rubisco is modified by chaperone protein?
Oh, actually, but this is interesting. Actually, I am not quite aware of this, because I can give you this idea, this part, particularly this part, because actually this molecular chaperone, it is actually associated with this assembly of this L8, S8. I mean, how they assemble. But actually it is a very beautiful question, and it is known. It is already in the textbook. Actually, I have not gone through this question. Actually,
okay, sir.
Because this is molecular chaperone. Actually, they assist in the assembly of this 8 large and 8 small. I would suggest, if you go through a book, Buchanan.
Yes. Yes.
In Buchanan, there is the actually this assembly is there, chaperone assembly, which can assist this assembly of this L8 and S8.
Okay, sir. Thank you, sir, for suggestion.
Sir, I have another question, sir.
Yeah, okay.
Yes, Rubisco take part in oxygenation reaction in C2 cycle, and similarly, Rubisco take part in Calvin cycle. In two cycle, Rubisco remain in same molecular configuration or change, maybe change. Molecular configuration of Rubisco.
Actually, what we know, actually, this regulation of Calvin-Benson cycle enzymes in C3 plant and C4 plant that are similar. I am not sure, but the light regulation of the Calvin-Benson cycle enzymes, both in C4 plant and in the C3 plant, they are similar. So if the regulation of Calvin-Benson cycle enzymes in C3 and C4 plant, that is in C4 plant, this Calvin-Benson cycle reactions takes place in the chloroplast of the bundle cell. So this regulation and the chloroplast of the C4 plant, this regulation by light is similar. So apparently you can predict, I am not sure that they are chemical, actually this structure is same, because this Rubisco is universal, whether it is in case of C3, C4 or CAM plant.
Yes, sir. When each take part in oxygenation, the Vmax may be changed, I think.
Where?
When each take part in oxygenation, and the
in
catalytic
case of C3 plant.
In case of C3 plant, but when it take part in Calvin-Benson cycle, the Vmax remains same. Is not.
No, Vmax or velocity of carboxylation, because there is the carbon dioxide concentration of mechanism is there. Actually, this here both carbon dioxide and oxygen is available, and the relative activity depends on the relative concentration. But actually, in the C4 plant, the bundle sheath cell where the carbon dioxide is formed by the decarboxylation reaction, actually in that case the oxygenase activity is minimum because of the low pH2 activity of the bundle sheath cell.
Yes.
Okay, low pH2 activity, because if you see the structure of the mesophyll cell and bundle cell, there is a distinct difference, because this mesophyll cell, because here this PP carboxylation reaction takes place in the cytosol, whereas the bundle sheath, this enzymes unique to this Calvin-Benson cycle that occurs in the chloroplast of the bundle sheath cell. Okay, so that's why there is a structural difference between this, because this mesophyll cell, it has the stacked membrane. So the stacked membrane is associated with the LH2 protein, and that is associated with the PS2 activity. But the bundle sheath cell has no stacked membrane. So that's why no LH2 protein. That's why no this PS2 activity. And this is one of the reasons for low oxygenase activity.
Yes. Thank you, sir.
Okay.
Participant, you have any question? Directly you can question to our.
I request participants, if any question in your mind, please ask Kumar sir directly.
One thing you can ask me, if there is any, because we are talking about this Calvin-Benson, but if the light regulation we can control.
I think, but in chat box there is no question. Arjun.
Hello.
Hello, Arjun.
Hello. No,
Uh,
no,
is
no
any
questions
question
in
in chat books?
No, 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 no,
No question.
no.
I think the session should be
You
closed.
can, yeah. You can
I am
conclude now.
possible. Okay. Thank you, sir.
Okay.
Professor Dr. Kumar Pal, for your good lecture as well as informative and effective lecture for our student and for our participant and also for us. And you have answered a lot of questions that will be to you from our participant. Now, from I again, on behalf of the Department of Botany, Egra S.S.B College and Prabhat Kumar College Contai, as well as both college, I express my gratitude to you, and if any question or any problem we have arising in future, I will contact you, sir. And if hello, sir.
Okay.
We have a program that after COVID period, after 7 or 8 months, both college or even organize a seminar. You should, sir. I request to you to give us the time, sir. After
Okay,
8 or 9 months.
Actually, you have expressed many things about me, but actually, I myself came myself as a teacher because I am very much